So today we are going to look at DNA replication and repair. So when Watson and Crick described the model of DNA as a double helix, they were also able to explain how the double helix will replicate. Okay. So they said when the DNA going from, say, 5' to 3', and this is 5' to 3'. So you have this sequence GATC, GATC, the nitrogenous bases in the middle. So if you have this sequence like this at the end, it is going to use this. This is CTAGTACG. It's going to use this template as the basis to replicate the new DNA. I'm marking that in a different color. So that is how they propose. This is how the DNA replication is going to happen. Okay, so here you have the new DNA strands are formed using the old template DNA. So this is also the template DNA. So in each strand, if you look at that, the new DNA is 1/2, the template DNA is 1/2. This is the old and the new. Okay. So this type of replication is called as a semi-conservative mode of replication. Semi-conservative mode of replication. So this is what they proposed, and but they did not provide any evidence as to how this could happen. And so you need an experimental evidence to really prove this is semi-conservative and not the other ways. So what are the alternative modes of replication? Okay. So alternative hypothesis or alternative modes of replication could be conservative mode. That means both will be old and the both strands of they will be new, or dispersive mode. Dispersive mode. So in the case of conservative mode, so if you have the sequence, a template GATC, and this side is a GATC, so this strand is going to be copied, like the template is going to remain as a template GATC, GATC, and the new strand is going to be GATC and GATC. This is another way the DNA replication, still utilizing the complementary base pairing that it can create. So this is the old, this is the new. That's a conservative mode of replication. So in the dispersive mode of replication, if you have this GATC and GATC here, this is the template. And this will separate into GT, and the same thing is going to happen like a GT. And let's put the A and C here, A and C here, and the same thing is going to happen as the CA, CA. So in each strand, you're going to have old and new mixed together. So this is considered as the dispersive mode, but the experimental evidence was to confirm this semi-conservative mode. Okay, So this is what is the correct one, and how it is proven by experiment is what we are going to get in now. Okay. So there are scientists Michelson and Stahl. So this is happening again in the 1950s after the DNA structure was described by Watson and Crick. Michelson and Stahl, they used stable isotopes. So what is the stable isotopes? Stable means non-radioactive isotopes N15 and N14. So these are the nitrogen isotopes of 1 neutron difference. Okay. So you may wonder, 1 neutron is just 1 AMU or Dalton, what difference it's going to make. But in reality, the DNA is huge, like 3.2 billion base per cell in human, or 5 million base pairs in E. coli, and there are gazillion number of those DNA molecules. It could be present in each, like culture of 5 milliliters, and so you have a huge difference between N14, N15, and N14. So this is the heavy isotope. This is the lighter isotope. Okay. So that is how you are going to separate. And they also, grew E. coli bacteria in this various medium containing N15, N15 or N14 medium containing medium, and the technique involved isolating DNA using a method called as cesium chloride density gradient centrifugation. So cesium chloride density gradient centrifugation will separate the DNA according to their density or mass per volume. So that's the technique they use. So something is heavy will be at the bottom of the tube, something light will be on the top of the tube, and so on. Okay. So when they started with, they had it all growing in N15. And so when they did the centrifugation, they found the DNA is at the bottom. So this is all N15, N15. It's all heavy at the bottom. Okay. And so they transferred the bacterium and then cultured in the N14 containing medium, and it's about 20 minutes. They allowed that to replicate in N14. So at that time, because it's a mixture of N15 and N14, so they had one strand of the DNA N15, the another strand is N14, and this had a combination of the heavy and the light. So this made a medium density band, Okay. So then they continued again another 20 minutes, and that is also in N14. And so in this N14 containing medium, the third sample had medium band of this N15 and the N14, and a light band of N14, N14. So this is N14, N14, and this one had N14 and N15. Okay. So that is how they found, so this is N14 and 15, a light band on the top and a medium band in the middle. That is what they found. Okay. So this is a first sample, this is a second sample, this is the third sample. Okay. So my question to you is, how did this prove semi-conservative mode and disprove the conservative and dispersive mode? Anyone can just speak up. And you can say the answer like, if it were conservative, I would see this, or if it were dispersive, I would see that, like that. Go ahead.
So I suppose it's semi-conservative because every single time half of the new DNA is from the new medium. So if it was conservative, the very first time you have, so after 20 minutes, you have a heavy band and a light band instead of a medium band.
At which sample?
The second sample. So after 20 minutes.
Okay. So if it were conservative, you would find a heavy band, both N15, N15, and a light band, right?
Right.
Yeah. So like this, both heavy, both light is what you would have found in the second sample. Good point. Anyone else want to explain why this is not dispersive, or how they disproved it is not dispersive? You can say, if it were dispersive, this is what you would expect to see.
Is it from the third sample that like when you split up like a dispersive DNA sample, it would still be medium in the third because it's a combination of both the light and the heavy.
Yes, exactly. So you would see a band mixed like this. So they did not see a dispersive. So this was semi-conservative, and both explanations are very good. Okay, thank you for speaking up. So if you look at this in an individual strand-wise, I'm using this black color for a straight line for N15. So this splits into two template, N15 and N15. And those templates are used to make the new strand of DNA. Let me use the same pattern of colors. So it is going to have N14 and N14 as the new strand being made here, half old, half new. That is what created that medium density band. Okay, let's continue the same logic. So when in the third replication, you have an N15 and N15, and the only two strands of N15, and the rest is all going to be this N14 and N14, N14, N14. So here, the new strand that is going to be made will be N15, N14, 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 and N15. So you have 2 or 50% of them are going to be the light density band, and this is going to be a medium density band. This is going to be a medium density band. That's a 50%. That is how they showed that this is semi-conservative. It's not conservative or dispersive. Okay. Any question so far? And it is not. You cannot say it's created voila, like from nothing. That doesn't happen either. That's not even considered. It's like magic, it appeared, so that didn't happen. Any question? Good. If you understood this far, so let's move on to the actual process of DNA replication. And so the DNA replication starts at origin of replication called ORI. Okay. So the origin of replication is a starting point where the proteins and the enzymes bind to the DNA. They cannot randomly start anywhere they like. So you need to have that origin of replication of a specific sequence, and the DNA is recognized. So in the case of prokaryotes, they have circular DNA, and there's a single origin of replication in prokaryotes. Okay. So this is where the single origin of replication, whereas in the case of eukaryotes, as all of you know, they have linear chromosomes, and they have the telomeres and centromere after they replicate, but the telomeres are there to begin with. So they have their longer and a much larger genome. So they have multiple origins of replication, multiple origins of replication. So the multiple RE here, it's a single origin. Okay. And we will see how the replication occur. And another thing to understand is the replication is bidirectional, so it will replicate. This is bidirectional, and this is also have a mechanism called rolling circle mode of replication. Rolling circle mode of replication, because after they replicate, they need to separate and become two separate circular DNA. Okay. So what happens at this point, it just opens up the DNA at the origin of replication. And this is referred to as a replication bubble. So we are going to put the orientation here, 5' to 3'. They go in opposite direction. That is how they can make hydrogen bond. Otherwise, they cannot make hydrogen bond. That's part of the deal. And inside is it's a hydrophobic pocket because of the nitrogenous bases. So the whole reaction works because of that, the hydrophobic nature of DNA. That's what keeps them together in addition to the hydrogen bond. Okay, so that needs to be kept separate. So this replication is going to happen, as I said, bidirectional. So that's what these arrows mean. It is going on both ways. And this entire thing is, you call this as the replication bubble. Replication bubble. And if you split that bubble in half, so this portion alone is called as the replication fork. So that includes from here all the way up to this point is a replication fork on this side, on the other side. So you can look at one at a time. So now I'm going to go through a series of enzymes in the sequence in which they work, and the enzyme and the function of the enzyme, and the image I'm going to draw on the right side. So keep following me. So I'm going to go through the first enzyme, is called as topoisomerase, and this is also known as gyrase. It's another form of gyrase. So this one relaxes the supercoiled DNA, relaxes supercoiled DNA. So if you remember in mitosis and meiosis, so this is at the G1 is where you would find here, this is a G1. And after they replicate, they go through this S phase, and then they go into this G2 and the M phase. So this is when you would see the chromosome, the replicated, the metaphase chromosome is visible only during this M phase, this metaphase chromosome. So during this S phase, the DNA is present as, anyone remember the term?
Chromatin?
Good, chromatin, chromatin. And the individual strands of the DNA, you know, is a chromatid. And the entire chromosome, like this entire thing, is a metaphase chromosome. So keep track of those terms. So now this one relaxes the supercalled DNA, goes from this chromatin into this double helix DNA, opens up like that. Okay. So that is the purpose of topoisomerase. That's how they function. Okay. So then the enzyme helicase unwinds the DNA double helix. So when it unwinds the DNA double helix, this is where the bubble is being formed. So now it opens up like this. It creates this bubble. And as I said, this bubble has hydrophobic pocket, and it will snap back with the hydrogen bonds and close this. It won't be like that. So this will be, this is how they're going to kind of create this helix again. And let's look at that way. That's the replication bubble is formed. And the next thing is single-stranded DNA binding proteins, single-stranded DNA binding proteins. So I should say the enzymes are the protein, enzymes/proteins. So they are abbreviated as SSB. They stabilize the single-stranded DNA. Otherwise, they will go back. Stabilizes single-strand DNA. So the SSB is going to kind of bind to this and hold the single-stranded DNA as they are SSB. So now we are to look at the primers are made by RNA primase. They make short RNA oligonucleotide. So this is a short strand of oligonucleotide. How it looks like, it's a 5' G, A, U, C, U, A, C, G, U, like that. This contains uracil instead of thymine. That's the RNA primer. And they will go and bind to this DNA. So this is where they would bind to the DNA to start the DNA synthesis, and we will see those primers. These are the RNA primers. Okay, so I'm going to keep track of that. So the next thing is the major one, which is DNA polymerase 3 comes in, DNA polymerase 3. And that one has 3 functions. Makes new DNA. So this is what is a DNA polymerase, makes a DNA polymer, and it proofreads, so it can go back and forth. It's a complex enzyme. And then if it finds a mistake, it repairs mistakes. So it's a single enzyme. It can do all these. Okay? So now I'm going to show only a part of this as a replication fork. And let's do this in a sequential manner. So this is where the replication fork is going to form. There's a 5' to 3', 3', 5'. And the replication starts at the 3' end of the template DNA. And this is the RNA primer. So that's the RNA primer. Okay. So the new DNA is being made starting with the RNA primer in this direction. The other direction has to go the opposite way. That is how DNA structure works. And so that is the new DNA being made. Okay, so this is the new DNA being made by the DNA polymerase 3. Okay. So at the beginning, everything looks same and so no big deal. But if you continue that as it expands, now you have more region open up, and whatever started at the beginning is going to continue, whereas whatever started here has to be restarted again, and this is made discontinuously. So one strand is made continuously, and that is happening in the same direction as the direction of replication. And so this strand that is made continuously is known as a leading strand. And this strand that is made discontinuously is known as a lagging strand. Okay. And the fragments of this lagging strand, there are 3 fragments. They are named after the Japanese scientist Okazaki. They call Okazaki fragments. Okazaki fragments. That's what the leading strand and lagging strands are made. So to continue on the same thread, you have the DNA polymerase 1 comes in, because now we have a mixture of RNA and DNA. This one removes RNA primer and replaces it with the DNA. So all these RNA primers here, and so all these RNA primer here, here, all of them are removed, and all these are RNA primers. They're all removed, and it replaces with the DNA. Okay. Then comes the next enzyme, DNA ligase. This one, it connects the DNA fragments. We saw these Okazaki fragments, and also the fragments on the leading strand with phosphodiester linkage. So this is how it makes this all complete phosphodiester linkage. The DNA polymerase cannot do that. It can only add 1 nucleotide at a time, cannot connect multiple fragments of DNA together. Then the last enzyme is the gyrase comes in, another like a similar to that topoisomerase. The DNA replication is complete, and now introduces supercoiling. So this is mainly to compact the DNA. So the cell is small, the DNA has to be compacted so that the chromatin is formed again, not the double helix is visible. Okay, we are done with the process. Any question up to this point before I continue? I'm going to zoom in on exactly what happens when the DNA polymerase 3 is creating this new strand, like we saw in protein synthesis and carbohydrate synthesis and so on. Any question up to this point?
There's no square cap today?
No, there was some issue. I will start it separately after the class. You will have the entire weekend to finish.
Okay.
Yeah. Any other?
Dr. Sada,
Yeah.
could you go up a little bit to where the cell cycle was?
Yeah, sure. So the G1 is where it's all the linear chromosome, but it is actually as chromatin. And S phase is where replication happens. The G2 is also chromatin. So all these are, G1, GS, G2, everything is chromatin. Only during the metaphases, you will see this mitosis phase, you will see the metaphase chromosome.
Okay, got it. Thank you.
Excellent. Okay. All right. Let's move on, and I'll hang around and I'll explain to you any question you have on the individual detail. So let's focus on the actual mechanism itself. So if you look at the replication bubble, so you're going from 5' to 3', and the other strand goes 5' to 3' in the opposite direction. And the 5', 3' is refers to the phosphate group. This is a hydroxyl group. This is a phosphate group. This is a hydroxyl group. Okay. So if you split this in 4, and the primers are added, and like this, these are going to be the leading strand, and the lagging strand is on the opposite direction of the direction of DNA replication. So when the new strand is being formed, so you have one side is leading, another side is lagging. So this is all leading, this is lagging, lagging, lagging. Okay. So that's how you need to see that. So these are lagging strands, and this is a leading strand. So this is a leading strand. Okay. So keep track of that. Now I'm going to zoom in on the DNA synthesis mechanism itself. So if you have a template DNA, it calls this G, it's called this A, and then T like this. And this is the 3' end, and this is a 5' end where this is going to start. And the G has C as the first starting nucleotide. So that is actually an RNA primer. And I'm going to put a single phosphate here. And this is going to continue like that. This has an OH and an OH. So this is part of the RNA primer end. This is where the 3' end of RNA primer like that. So the next DNA that is going to come here, I'm going to put that is a new DNA being made. This is the T, and so that is going to have deoxyribose, that's a DNTP, hydrogen hydroxyl group. And this one has this phosphate. I'm not expanding the phosphate. It is PO4. And so what happens here is there is nucleophilic attack that happens. This is not condensation synthesis, nucleophilic attack. That's how the DNA is going to be connected. And these two will come off as PPI, or that stands for pyrophosphate, this inorganic pyrophosphate. They will then become 2 inorganic phosphate will be used to make this DNTPs again. Okay, but if you were to look at the number 1', 2', 3', 4', 5', same here, 1', 2', 3', 4', and 5'. So this will result in this reaction. You would find that will result in, I'm going to put that here. You have the oxygen connected to this phosphate. This is a negative strand, and here's this double-stranded oxygen. This oxygen is connected to the sugar on the top, and this is the sugar at the bottom. So this is the 3' end of the sugar. It's a 5' end of the sugar. This is what you call as phosphodiester bond or linkage. Okay, that's how it creates. So the next DNTP is going to come, and this will be an A. And this also, let me draw that next sugar is a hydrogen. So this is a DTTP, and this is DATP, and so on as an OH group. And you have this phosphate, phosphate, and phosphate. And the same thing happened here. Nucleophilic attack happens here. These two will come out as a pyrophosphate, PPI. That's how it is going to continue. And this will become this phosphodiester linkage. And this is the sugar 1. and sugar 2. Because the phosphodiester is a linkage between an alcohol and an acid. There are 2 ester linkages. That's why it is called phosphodiester linkage. So if you see that the way it is happening here, I'm going to keep track of this 1, 2, 3, 4, 5. So this is the end point. This is the starting point. So if you see this, the replication of the DNA happening in the 5' to 3'. So this is happening the opposite direction of 5' to 3'. So this is that 5' and this is that 3'. I hope everyone understand when we say replication happen 5' to 3'. Okay. Last but not the least, I will wrap it up by the DNA repair mechanisms. And even if it takes a little bit longer, but let's complete the whole story. The first repair happens is a mismatch repair. And this happens during DNA synthesis, during DNA replication, and the enzyme DNA polymerase. So DNA replication happens in the S phase, DNA synthesis phase, and this is done by DNA polymerase 3. That has the synthesis ability, proofreading ability, and repairability. The next to repair is an excision repair. And this happens after the DNA replication. And this can be in G1 or G2 phase. Any damage happened to the DNA, this involves endonuclease that will cut the damaged part, and then DNA polymerase 2 and DNA ligase. All these will do that excision repair. The last but not the least is telomere repair. So the telomere repair is a little bit tricky in the sense during S phase, the enzyme telomerase will protect the telomere from degradation. Why the telomere is degraded? See, for example, if this is a 5' to 3', so this is the 3' end is a telomere region, and you add a RNA primer here. This is the RNA primer, and you make the new DNA strand from starting from this. The new DNA is being made. After the new DNA is being made, the RNA primer is degraded. This part is degraded. So the telomere becoming shorter and shorter. This becomes single strand. RNA has to go because of this DNA polymerase 1 will come and remove that. So with telomerase, what it does is it extends that 3' end. This is the telomerase enzyme, will give a fragment of RNA. It will create this extension. So this is the extension of the template strand. Then the primer will bind to the extended region, and the new strand is being formed from the extended region. After the RNA is removed, the extension is also removed. The length of the telomere is preserved. That's it. So that is happening in young and actively growing cells, as well as in cancer cells. The telomerase is active. That's what makes the cancer cell immortal. All the other cells, the telomere becoming shorter and shorter and shorter, they stop dividing and replicating anymore. Okay, so that completes it. And